moving on to sex differentiation. Now, what is sex differentiation? How does a subject develops his sexual identity, and how does there is a growth of reproductive structures? When it comes to this sexual differentiation, we can study it in 3 levels, doctors. Number 1, genotype. By genes, how they differ. By gonads, how do they differ. By body, how do they differ. Alright, it is a genotype sex. It is called as genotype sex. Number 2, it is going to be gonadal sex. And number 3, it is going to be phenotype sex. Why is this so? Can we put all these things under one cluster and simply say differentiation of sex? No. When it comes to abnormality, subjects may have an absolutely normal genes, but they may have a abnormal gonads, because the conversion of gonad from an indifferent one to be a precisely male gonad or female gonad is not just dependent on one gene. We just discussed, right? If someone has got a SRY gene, that SRY gene is going to produce a factor called the testis determining factor, and this testis determining factor is going to promote growth of the gonad into a male kind of gonad, and all the hormones are going to be male pattern of hormones, isn't it? So, is it all about growth of the gonad? No. We have plenty of transcription factors coming into the picture. We have multiple accessory gene products coming into the picture to develop a proper gonad. So, someone may have a absolutely normal genes, but gonadal growth can be compromised. Or someone will have a normal gene and normal gonad, but the other ancillary growth of male pattern or female pattern might be compromised. Alright, because there are going to be abnormalities in 3 different ways, and these abnormalities are handled, diagnosed and treated in 3 different methods, we are going to categorize the differentiation into 3 clusters. We are not going to put everything under one pocket, one umbrella, saying that this is how someone is going to differentiate sexually. It is not just that simple, people. So, there is a gonadal level of differentiation, there is a phenotype level of differentiation, and the fundamental genetic level of differentiation, something you are already familiar with, right? Now, let us get to see how does these kinds of differentiation takes place, beginning with genotype. Now, when it comes to genotype, guys, it all begins with fertilization of gametes, right? I hope you know the difference between a normal cell, a somatic cell, and a gamete. A somatic cell is diploid, right? Which means we can call it as 2N. 2N refers to diploid, guys. This one. 2N refers to diploid. But when it comes to a gamete, it is haploid and it has just got 1N. So, what is this 1N? What is this 2N? N refers to set of chromosomes from one parent. If I take a cell, that cell will have 2 sets of chromosomes. One set comes from the dad and the other set comes from the mom. Alright, so these 2 sets of chromosomes are going to be present in single cell of the offspring, single cell of the child. So, this cell, since it has got the 2 sets, identical 2 sets, one from dad and one from mom, this cell is called as a diploid cell. Alright, but when you take up a gamete, alright, for the same child I am talking about, when you take up the child's gamete, that child's gamete will either have a pattern of chromosomes with, of course, there is going to be meiotic process going to take place, there is going to be, changing of genes between chromosomes itself, that's on one side. But when you count the number of chromosomes in a gamete of a child, the number of chromosomes are going to be just 23. It is not a pair, it is not a set, rather it is going to be just one set of 23 chromosomes, it is not a paired one. Since there are only 23 chromosomes, 22 out of which is going to be autosomes and 1 out of which is going to be sex chromosome, alright. That may be X or that may be Y, it doesn't matter. But when you take up a gamete, it just got one set of chromosomes, just 23 in number. When 2 such gametes fuses, so this one haploid cell gamete and another one haploid cell gamete, when they fuse, now there is going to be 23 pairs, alright. Now there is going to be 46 chromosomes, and hence that is going to be called as a diploid zygote. So, this all differentiation begins from fertilization, and when it comes to this word fertilization, there is going to be fusion of 2 haploid gametes into one diploid zygote, guys. Alright. So when I say two gametes are fusing, what are all the sex chromosomes present in these two gametes? Is it X chromosome and X chromosome, or is it X chromosome and Y chromosome? So people, when it comes to that word, the sex chromosome contributed by parents leads to genotype of the individuals. So genotype sex of the individual is determined by sex chromosome, and who contributes to that sex chromosome? It's the parent. As I was telling you, male gamete would be bringing in one sex chromosome, maybe X, maybe Y. Female gamete would be bringing in another sex chromosome, definitely X. So based on the arrangement, is it XX or XY? We are going to identify by gene someone as male or female, and that is what is called a genotype sex, doctors.